Today, we're going to be talking about data analytics that relies not just on experimental data, but is enriched with various knowledge types to help elucidate biological functions and properties. So I'll talk just for a few minutes. I'll be followed by Errol, who's going to talk about the use of knowledge-based classifiers, and then by Jonathan Monk, who's going to talk about pan-genomics and how to get at strain-specific properties. And finally, we have a more recent topic from Anand, which is a new way to analyze bacterial transcriptomes. So this is a graphical summary of the talk. So Errol will be talking to us in panel A, where we are trying to find separating planes in high dimensions to separate resistant strains from susceptible strains based on binary phenotypic measurements. And what's unusual here is that the axis of this space actually represents mechanisms. So that plane represents an integrated cellular function, as I'll show on the next slide. John did a pioneering paper back in 2013 on the first mechanistic analysis of a few dozen E. coli strains, and he was able to predict metabolic capabilities that led to this notion of metabolic classifiers, and he was able to classify these strains and match them up with colonization sites. And pan-genomics has advanced notably since then. And then finally, panel C represents what Anand will be talking about, and that's to use signal extraction algorithms from a complex signal to identify sources. And we've all heard about microphones being spread out through the stage of an orchestra, and algorithms applied to isolate the sound from individual instruments. And that's the signal extraction algorithm approach. And we're using this for transcriptomes that we can maybe view as orchestrated set of responses of multiple transcription factors. And if you have enough points in a space of experimental conditions, you're able to extract the signal that's associated with transcriptional regulators. So this slide here illustrates what I mean by having mechanism in that separating plane. This is going to be done by flux balance analysis, so that plane represents some metabolic process, like the one shown to the right. So we are not looking at alleles and associating alleles with phenotypes, but we are now looking at associating a whole cellular process that is underlying an AMR resistance phenotype. As I said, John had a pioneering paper here in 2013 on a pan-genome analysis with a few dozen strains. Since then, we've been analyzing a few hundreds and a few thousands, and with every increase in order of magnitude, we are learning new things. When we got to the thousands of strains, something called allelomics emerged. We were not just looking at presence-absence calls of genes in a genome, but also what particular allele that strain had of that gene. And that leads to classification of, for instance, protein structures as shown in that little image. And we are now about to move on to 10 to the 4th, tens of thousands of strains, and we anticipate there will be new lessons learned there as well. And finally, Anand is going to talk about transcriptomics. And this illustrates one of the lessons we have learned from his approach. And that is that as we build up a database of expression profiles and start putting in new data from individual studies, we learn new things from the data we didn't ask before. So in other words, these 4 studies indicated there were all carried out to answer specific questions that required generation of expression profiling data. But when that's put into a bigger database and you have these signal extraction algorithms operate, you will find new information in these profiles that weren't obvious from the previous compendium of profiles. So that's the introduction. I'm going to hand it over to Errol now. Errol is a senior graduate student. In fact, he is graduating and defending on Monday. And he had a beautiful paper in Nature Communications on his method that came out, I think, 2 weeks ago that he's going to tell us about. Errol, take it away.
All right, well, thank you for the introduction there, Dr. Paulson. My name is Errol, and I'll be talking about these knowledge-based classifiers which integrate flux balance analysis with GWAS data. The motivation for these approaches really comes from the pathogen data deluge that we are currently experiencing. Specifically, there are thousands of pathogen clinical isolates sequenced. And so the figure below shows a search on the Patrick database for Mycobacterium tuberculosis in 2018 and 2019. And so there are tens of thousands sequences available. These strain sequences also have corresponding AMR phenotypes available. So we know whether these strains are resistant or susceptible to a wide range of antibiotics. So the goal here would be to leverage this data deluge to extract insights into antimicrobial resistance, both genetic and metabolic. So the first approach did not use any knowledge. This was a pure machine learning approach. We did this back in 2018. We used a support vector machine equipped with some regularization, bootstrapping, etc. But we showed that this approach was able to identify 33 known AMR genes and also propose 24 new AMR genes. And this greatly improved the identification of AMR genes over just common GWAS methods that are univariate tests. However, there's nothing in that approach that can really tell us about these alleles. The SVM and these machine learning approaches are fundamentally limited because they don't really say anything about the function of these genes. The SVM has no idea what a protein, a metabolite is, etc. And so the goal here would be to use a metabolic model of tuberculosis, this genome scale model that it captures thousands of genes and their metabolic functions, use this model under this flux balance analysis framework to somehow classify AMR, and then interpretations would hopefully be interpretable on the mechanistic level. So this is a comparison of the approaches. On the left, we have the support vector machine, and on the right, we have the metabolic allele classifier, which is this knowledge-based classifier that integrates the model. So on the left, we could see that we have the strains colored red and blue for resistant and susceptible. The SVM basically identifies this plane that splits between the red and blue points as well as possible. And then we can interpret this plane through the coefficient C to identify the allele. So that's what we did in 2018. Now, what we show through this metabolic allele classifier is that we could transform these strains to this metabolic flux space in such a way, along with this classifying plane, where now the model, when we interpret it, not only do we identify alleles, but we could also get their corresponding biochemical states. So how well does this metabolic allele classifier do in comparison to these common machine learning approaches? So we find that they actually perform on par with the support vector machine as far as prediction accuracy, which has been usually a limitation for genome scale models. And so in the figure to the left, or bottom left, we see that we get area under curves for these receiver operator characteristic curves around 0.93 to around the 0.8 and 0.71, which is quite good for these antibiotics. Not only that, but they could also identify known AMR genes. And so to the figure to the right, we can see that the highest weighted coefficients correspond to the primary AMR genes. And this example is for pyrazinamide. So I'm going to quickly go over two examples here for two antibiotics and show what kind of deep insights we get. So application to pyrazinamide AMR. So we find that when we apply this metabolic allele classifier to pyrazinamide, we're able to recover the known genetic determinants, PNCA and PPSA, and we also implicate ANSP2 as a new AMR gene. So the unique thing about this approach is not only do we identify these genes, but we also, it provides estimates for these allele-specific flux effects. And so for the figure to the bottom left, for the PNCA alleles, we see that the two alleles found in predominantly resistant strains are estimated to be loss of function, which is consistent with the literature on PNCA, which are actually usually deletions. not only do we find these allele-specific effects, but we could also see pathway-level flux states discriminating between resistant and susceptible strains. And so when we do some enrichment with these fluxes on the pathway level, we could see that we're able to explain AMR using flux through nicotinamide metabolism, as well as thioceryl metabolism. So these are interesting because they actually recapitulate what is known about pyrazinamide. There's a paper in 2016 that identified thioceryl metabolism as a mechanism of pyrazinamide resistance through CoA regeneration. We also hypothesize ANSP2 alleles confer resistance through modulation of coenzyme A. And so that's a completely new hypothesis identified purely computationally. For another drug, para-aminosalicylic acid, we identified Thi-A and Cis-K2 as AMR genes. Thi-A is a known AMR gene, while Cis-K2 is a new one that's implicated. We estimate the resistant dominant Thi-A alleles to be loss of function, as well as the Cis-K2 alleles. On the pathway level, we identify folate metabolism, as well as sulfur metabolism through cysteine and methionine as the key discriminating pathway fluxes determining pass resistance. When we go deeper into the network, we see that these Cis-K2 alleles that are associated with resistance are contributing to this predictive fluxes through modulation of L-homocysteine. And so that's a completely new hypothesis provided by our model. The identification of cysteine and methionine metabolism too is consistent with a paper in Science in 2013 that identified that pathway as the most altered flux pathway bypass activity. So in conclusion, we developed a biochemically interpretable machine learning model that integrates a genome scale model. And then we show that interpreting this model identifies both known and new AMR genes, and also provides biological explanations about their metabolic flux states discriminating AMR. So I'm done with this, and I'll hand it off to John Monk, who is a leader in this field of microbial genomics. Thank you.
All right, thanks, Errol. Can everyone see my screen?
Yes.
Okay, great. So I'm going to go and talk now about concepts of the pan-genome and how we're developing new methods to analyze the pan-genome. To start, I'd like to talk just a little bit about what is the pan-genome. So the pan-genome is really the totality of genes found amongst members of a species. And a pan-genome can be either open, which means that every time we sequence a new strain, we start to find new genes that weren't found in previous sequence strains, or it can be closed, which means that a lot of newly sequenced strains really just contain combinations of previous genes that have been seen. And you can classify the pan-genome, the size of the pan-genome and the shape of the pan-genome using this mathematical formulation called Heap's law. And that basically just looks at the shape and size of the pan-genome as it increases over time. And that actually came from linguistics, where they were actually looking for mathematical methods to analyze the complexity of text. And you could look at a book and say, how many times do I see new words as I go through chapters of this book? And so you can kind of think of the pan-genome as a similar concept. How complex of is a species? How often do I see new genes every time I sequence a new strain? In contrast to the pan-genome, there's also something known as the core genome, which is just the number of genes that are shared amongst all strains in a species. And that's represented by the green curve on the bottom there. This concept of a pan-genome is becoming more and more appreciated, but it was really first noticed back when the first strain of E. coli was sequenced, and that was E. coli K-12, which is the common lab strain. It was sequenced in 1997, and when that happened, people thought, hey, we have an E. coli genome, we can go on and sequence other organisms. But then in 2001, E. coli 0157, which is a pathogenic strain of E. coli, was sequenced, and people were shocked to realize that it actually had 1 million more base pairs than E. coli K-12, and beyond just the larger size of the genome, it turned out that only about 30% of the genes were shared between these two different strains. And so that started to indicate that really there's a lot of diversity amongst different strains of E. coli. This realization and further sequencing of E. coli strains led us to realize that we needed to expand our metabolic network reconstructions to cover the diversity of different strains in the E. coli species. And so we actually took this well-annotated, high-quality metabolic network reconstruction of E. coli K-12 MG1655 that was actually developed in our group over the last, at that time, 14 years, but now it's been over 20 years, that accounts for the function of 1,366 metabolic genes, over 2,200 different metabolic reactions, and that participate with 1,100 unique metabolites. And that genome scale model was very high quality, but it was very specific to E. coli K-12 MG1655. And so what we did is we actually selected 55 different strains of complete E. coli strains, as well as Shigella that spanned different pathotypes, be they commensal or healthy strains that are not pathogenic, intestinal pathogens, or extraintestinal pathogens. And we mapped metabolic content from E. coli K-12 and 3 other curated enterobacteria reconstructions, including Salmonella, Klebsiella, and Yersinia. And we also added further specific information based on the literature to build these genome scale models of 55 different strains of E. coli. And from that, we were able to then classify what is the shared and unique content amongst these different models of E. coli. And this is an example of really enriching the pan-genome with different knowledge types. So in this case, we actually had metabolic reactions that we could layer into different subsystems of metabolism and study how are these strains similar and different. And so amongst the 55 different strains of E. coli, the core number of metabolic genes was 965, while the pan number of metabolic genes was 1,460. And you can see how that was stratified based on different reaction subsystems. So in the red, the core metabolism was really conserved in some specific areas of metabolism, like energy production and conversion, nucleotide metabolism, etc. However, carbohydrate metabolism was far less conserved, and there are a lot of different genes that fell into this kind of pan metabolism. The power of these genome scale models is that we can actually go from what's known as a reconstruction into a actual functional model. So we can use flux balance analysis, which Errol mentioned briefly above, to basically calculate phenotypes based on the structure of the network in these different strains. And what that allows us to do is actually predict the ability of these strains to grow in hundreds of different nutrient environments. So the figure on the left is just demonstrating that we can go from strains and their genomes to these in silico models that can then be used to actually predict phenotypes. And what's exciting about this is that you've now gone from this kind of concept of a pan-genome to specific testable hypotheses about what distinguishes these different strains. And so on the figure on the right, you see a cluster map of predicted growth and no growth capabilities for the 55 different strains of E. coli and Shigella. And you see that they actually cluster by these different pathotypes. So Shigella strains, you can see, have lost the ability to grow on a lot of different nutrient sources, whereas extraintestinal pathogens are distinct from the commensal strains and the intestinal pathogens. And from here, you could actually perform further analyses to determine what are the exact nutrients that are actually distinguishing these strains and how do they map on to different colonization sites or nutrient niches in the body. And this paper here we published in 2013 really demonstrated that metabolic capabilities are very different for different E. coli strains, and they can be used to predict colonization site from sequence alone. The models also were able to predict auxotrophies for cases of pathoadaptation to different hosts and environments. So that's a common case in Shigella, where they have what are known as these black holes in the genome, where a lot of the strains are missing specific biosynthetic capabilities. And actually, if you start to look at why those may be missing, some of them are related to the virulence and the lifestyle of those strains. And ultimately, we could predict infectious niche from these models. So based on that study, we've now applied similar tools to study a range of different organisms. One of the next studies we did was actually studying Leptospira. So we looked at actually 4 different species of Leptospira, some of which are pathogenic and some of which were saprophytic. And we found actually that building these genome scale network reconstructions, the ability to synthesize vitamin B12 was limited to some of the pathogenic species in Leptospira. And this may have given them a survival advantage in the human host where B12 is sequestered by the body. We've continued building genome scale network reconstructions for other organisms, including Staph aureus, and we've also been layering on additional types of information. So we built these genome scale models for 64 different strains of Staph aureus, and we also looked at the number of different virulence factors identified amongst those genomes, and that kind of added new information. So now we have predicted metabolic capabilities as well as presence or absence of different virulence factors. And that was useful in understanding strain diversity amongst Salmonella strains, where we showed that we could predict strains host based on presence of different toxins, adhesions, as well as metabolic capabilities. We've also expanded our studies to study E. coli in a deeper set. So as Errol mentioned, there's this data deluge and more strains are being deposited every day. And at the time this paper was published, there were about 1,000 different strains or 1,200 different strains of E. coli that are available to study. So we extended our approaches from the 55 strains to these 1,200 different strains and built genome scale models based on that. But we also started to classify not just the actual presence or absence of different genes in the pan-genome, but actually the diversity of the alleles encoding those genes. And we could look at, which sets of metabolism, which pieces of metabolism have more conserved alleles versus more diverse alleles, and try to learn something about whether that tells us anything about the evolutionary pressures on those genes, if it tells us anything about the lifestyle of those genes, or those strains, et cetera. And we also scaled up our analysis to study intestinal pathogens and extraintestinal pathogens, and showed that a lot of the different carbohydrates that we found were still distinguishing amongst those different strain types. Beyond just looking at gene presence absence, we've also started to look at the actual structure, the context of genes on the genome. So in this case, looking at the structure of the AGR area on the genome, and we found that AGR type and the actual number of base pairs between genes in the AGR region can be used to predict virulence factor profiles, and that also divergence in this histone kinase protein confers different signal specificity amongst strains of Staph aureus. We've also studied specific subsets of strains. So in this case, looking at E. coli strains that are known to cause or implicated in causing IBD. And in that case, a lot of strains in the B2 clade of E. coli are known to be associated with IBD. And we looked at building reconstruction of those strains, and we found that a lot of these B2 strains were unique compared to other strains of E. coli based on their ability to catabolize sugars derived from mucus glycan, particularly this tagatose metabolite. And so that really motivates the study of the nutritional effects in these strains and how that affects their pathophysiology, particularly for IBD patients. We've also expanded our studies to strains of Salmonella. And we've actually looked at pan-genomes of different serotypes and tried to understand if there's certain sets of genes or certain sets of metabolic capabilities that track along with the different serotypes and pathotypes of Salmonella strains. And we showed that certain metabolic capabilities could be used to distinguish these different serovars and serotypes. And of course, that hints that these host ranges are limited by the metabolic capabilities of different strains. And we've also mapped, now we've gone beyond just conservation of these genes, but actually mapped structural features of based on 3D structures of proteins onto the presence of these different genes and their different alleles, and try to understand what that can tell us about reactive oxygen resistance among strains in the species. And we can use genome scale models to also measure the effects of ROS in these strains and simulate the different production of ROS to show that antioxidant properties are exhibited in the structural proteome. And so I hope I've shown to you that these methods can be applied to a range of different organisms, and they're robust to different sizes of data sets. And that's important because if you just plot the number of sequences that are available for a lot of these organisms that we work with, including E. coli, Staph aureus, Salmonella, Acinetobacter baumannii, as well as many others, the number of sequences that are available is actually doubling. And it's growing exponentially, and it's doubling at a rate of about every year. And so obviously that's very important because we're getting to this point now where we can analyze tens of thousands of strains in a species. And so we've gone to the point where we'll learn hopefully new lessons from these different sets. So really the key takeaway from this section is that the structure of the pan-genome is really responsible for strain phenotypes, properties, including their lifestyle, virulence, and antibiotic resistance. And this accessory genome, the difference between the pan and the core genome, the accessory genome drives the diversity amongst strains in a species. And we can use that to really explore specific features of these organisms, like their metabolic capabilities, auxotrophies present in certain strains, how that maps onto their nutrient niche. We can go deeper into the different presence and absence of genes and looking at sequence variation. And then we can even map that around the world and understand that there's certain subsets of strains that are present in certain areas, or are they evolving differently in different parts of the world, for instance. And this is really important because as more and more strains are sequenced, we'll need tools like this to apply to those strains so that we can learn useful information about them. And I think it's important to note that this exponential growth of genome sequencing projects means that in 1 year from now, we'll have as many additional data sets to analyze as currently exist. And so hopefully that sinks in and we realize that this data deluge is really a challenge, but an exciting challenge to pursue. And so with that, I will pass it on to Anand. Anand is a postdoc in our group who recently finished his PhD here, and he's going to talk about some exciting methods for analyzing transcriptomes.
So transcriptomics data sets have also been exploding in recent years, especially RNA sequencing data sets. So if you can see on this image on the right, the transcriptomic data sets that are available for E. coli MG1655 have been exploding over the past 5 years. And about 1/5 of that data set was actually produced by our group. So this data was published in 15 different publications spanning 5 years and all used the same standardized sequencing protocol. So I decided to take all of this data, put it together into a compendium that we named PRECISE. So PRECISE contains 278 different expression profiles spanning across over 100 unique conditions. And this is the largest single source RNA-seq dataset for any bacteria. And as the name suggests, this is a very high quality dataset. So in order to get some sort of biological understanding from this dataset, we turn to machine learning, or specifically independent component analysis. So ICA is a blind source separation algorithm. The way that it works can be demonstrated by an orchestra. So you imagine an orchestra on a stage and they're all tuning up, and you put a bunch of microphones in this stage. Each microphone will pick up a different linear combination of each of the instruments depending on which one it's closest to. We can then use ICA on this combination of mixed signals to get back the original source signals or the original songs that each instrument is playing. And we also get the relative volume of each of these individual instruments on each of the microphones. So if we go over here to the biological side on the right, each transcription factor in a bacteria is sending out a signal across the genome, telling certain genes to be upregulated and certain genes to be downregulated. So it's the combination of all of these different signals from these transcription factors and sigma factors that give you the expression profile under a certain condition. So we hypothesized if we take enough unique different conditions and collect them together, can we apply ICA to that and get back the original source signals or transcription factors, binding sites? And can we also get back the relative activities of each of these transcription factors under each individual condition? So we applied ICA to PRECISE, our RNA-seq compendium, and got back 92 source signals. So after processing these signals, we got back what we call imodulons. So an imodulon is an independently modulated group of genes. This term is borrowed from the word regulon, which is the group of genes that are regulated by the same transcription factor. So an imodulon represents an expression signal. For example, all of the genes that are affected by a specific transcription factor. So there are a few properties about imodulons that we get from ICA. The first is the gene weightings. So over here, you can see that this is a specific imodulon, and you can see that there are some genes that have very high gene weightings, such as GLPD. And this tells us that the effect of the underlying signal or the underlying regulator behind the signal has a very high effect on GLPD. So GLPD expression changes a lot when this signal changes. However, GLPX doesn't have as strong of an effect. And you see that there's about 4,000 genes here that just don't change when you change the underlying signal. So we can take all of these high-weighted genes and compare them to regulons that are defined in the literature. And in this case, we see that all of these genes perfectly overlap with the known regulator GLPR, which has to do with glycerol uptake. So we also get activities for imodulons, and this represents the overall expression level of genes. So you can see over here, there are some experimental, each bar here represents a different condition in our data set. And there are some experimental conditions that have very high activity for this specific imodulon. That means that these genes are highly expressed in this imodulon, or sorry, in this experimental condition. Whereas down here, when you have an activity near zero, that means that the expression of these genes is similar to that of our baseline condition, which is glucose minimal media. So the way that this imodulon works is you multiply this GLPD. For example, if you want to know the expression level of GLPD under a certain condition, you multiply this gene weight here times the activity under that condition. And you do that for every single imodulon. Now, most genes only exist in one or two, usually one imodulin, but it's the sum of all of these changes that gives us an approximation of the gene expression level. So here's an example of an imodulin in action. This is the PURE-R1 imodulin, and it's shown down here. So all of the genes here are related to purine biosynthesis. You can see that this is the purine biosynthetic pathway. And there are some genes in this imodulin that have positive coefficients, which are indicated in blue, and some that have negative coefficients, which are indicated in red. This gene here has to do with purine degradation, and this gene here was previously uncharacterized. So these imodulin activities reflect the overall expression of these genes. You see that when we add exogenous adenine to the system, the cell realizes that there's too much adenine and it downregulates this purine biosynthetic pathway. At the same time, it upregulates purine degradation and this uncharacterized gene. On the other hand, if you knock out PURE-R, then that increases purine biosynthesis because it's no longer being repressed by PURE-R. And again, this is all in comparison with 0, which is our growth on glucose minimal media. So based off of this information, we were actually able to characterize YDHC as a novel adenosine or adenine transporter using a few biochemical studies. So this shows how imodulins can be used to get new information on such a highly characterized organism like E. coli MG1655. In addition, we found a similar imodulin in a staph data set. And this imodulin's activity was highly correlated with a virulence factor. So as I showed in the previous slide, each of these imodulins has its own unique story. Each imodulin has certain genes that are present in it, and we can compare those to the static regulons. So regulons are static lists of genes. They're just all the genes that are known to be regulated by a transcription factor, versus imodulins are a dynamic data-driven set of genes that we also get this dynamic activity profiles to understand how these genes are affected by different environmental conditions. So all of these imodulins are shown here that are related to different transcriptional regulators. We're going to focus in on RPOS, which is the largest imodulin. It contains over 100 genes. So RPOS is a sigma factor. It's part of the RNA polymerase complex, as you can see up here, and it's specific to activating genes that are related to stress response. So the RPOS imodulin indicates the cell's stress level. When you have stressful conditions like these 4 shown here, RPOS imodulin is upregulated. So all the genes that are regulated by RPOS are upregulated. On the other hand, if we evolve E. coli for fast growth, we end up accumulating mutations in RNA polymerase that downregulate the RPOS imodulin. So all of the genes in the RPOS imodulin have lower expression compared to the wild type. Now there's another imodulin, this translation imodulin, that contains a lot of genes that have to do with ribosomal subunits or other translational machinery. And we see that there's a correlation in the activities of the RPOS imodulin activity and the translation imodulin. Down here, you can see that there's a very high amount of stress expression, low amount of ribosome expression, and very low growth rate indicated by the filling of these points. Up here, you have a very high growth rate, lots of ribosome expression, and very little stress expression. And we actually identified a few mutations in RNA polymerase that shift strains up and down this global trade-off. So we can also use imodulins in place of differential expression analysis. And this reduces our analysis from looking at 4,000 different genes to just 90 imodulins. So this is another example of a mutant RPOB strain. So these 2 strains only differ by a single mutation in RPOB. And we did expression profiling of these 2 strains and found that there was only 13 genes that were differentially expressed. However, I've also marked on here, all the genes in red are part of the RPOS imodulin. And you can see that there's a small, subtle, but still consistent shift down in the expression in the mutant strain compared to the wild type. This is not, we are not able to detect this using differential expression analysis. However, over here, when we do differential imodulin analysis, it is very clear that RPOS and GAD-EWX are the 2 transcription factors that have different transcriptional effects from these 2 strains. And so these 2 imodulins control over 100 genes. And the only way that we were able to capture this small shift is because differential expression analysis only compares 2 conditions, versus differential imodulin analysis uses shared information from hundreds of data sets to characterize these different strains. So we can also expand our imodulon computation to other strains of E. coli. So here we grew 8 E. coli strains on the same identical condition, again, glucose minimal media, and we inferred the imodulon activities for each of these 8 strains using the transcriptional signal from MG1655, our baseline strain. So we found that RPOS had the most varying activities, even though it was always grown on the same condition. So we looked into the genotype and found that the strains that had the lowest RPOS activity had a stop codon very early on in the RPOS protein. Then we had some strains that had a glutamate and a glutamine, and those also seemed to change its activity. So we have a clear connection between the genotype and phenotype. The mutations in RPOS explain the imodulon activities. So we've also expanded our ICA computations to other organisms. For example, we created a precise or an RNA-seq compendium for Staph aureus USA 300. So this is a hospital-acquired MRSA strain, and we collected over 108 different experimental conditions, and we're able to pull out 29 imodulons from this data set, things ranging from metabolic to stress response to virulence factors. So there's a progenitor strain for USA 300 that's known to be much less virulent than USA 300. And this HG001 strain is very similar to that precursor. So we also computed imodulons for this strain and found that there was one imodulon related to the AGR virulence factor that shared quite a few genes between these two strains. However, these genes over here in blue are only present in the USA 300 AGR component and not in the HG001 imodulon. These genes here have to do with biofilm formation and alpha toxins. And so this shows us how imodulons can give us hints at what's going on in the regulation and why different strains have different virulence. So until now, we've been focusing on imodulons from individual data sets, all from the same research group. But we wanted to know, can we use this as an integrative tool? Can we take data sets, for example, here, 2 RNA-seq data sets from different groups and 3 microarray data sets that use different platforms? Can we combine all this data together and can we get imodulons from that? And what we found is when we did ICA of each of these data sets by themselves, the imodulons were strikingly similar across all of these. And when we combined them together, we also got imodulons, and we were able to characterize a new imodulon that was actually regulated by PPGPP, which is a small molecule alarmone. So this shows us that not only can ICA capture transcription factors, but any signal that changes the global transcriptional structure across the cell. So this shows that ICA can extract actionable biological knowledge from disparate data sets. So with this knowledge, we turn to the NCBI SRA. So this is a database that has tons of RNA-seq data for all sorts of different organisms, and we're starting to build imodulons for each of these organisms. So we already have imodulons for E. coli, and we compared them to the imodulons we got for Salmonella, and found that a lot of them are strikingly similar. So the genes that are in these imodulons are almost identical, and they're regulated at the same ratios. Down here, when we get a little farther away from E. coli, Acinetobacter baumannii has a similar imodulon to E. coli, and these are both FER. This is the ferric uptake regulator, has to do with iron. And we see that the activity of this imodulon actually drops when we treat A. baumannii with azithromycin. And so this is giving us an interesting observation about the physiology of this bacteria. And so imodulon knowledge can be used and shared across different data sets and organisms to help us characterize the transcriptional regulatory networks for some of these less characterized organisms. And all of this data is already available. It's just it just needs to be used. Imodulons offer this dynamic low-dimensional interpretation of the transcriptome. And imodulons can reinterpret data that already exists and answer questions that weren't answered or even asked in the original study. All right, now we can turn it back over to Professor Paulson.
Thanks for that. Those were 3 just nicely illustrated segments on how we are trying to map knowledge onto data sets to learn much more from them than we would from the data alone. And you could maybe think of this as a combination of top-down and bottom-up approaches, the top-down being the broad data sets and the bottom-up approaches being the mechanisms we're trying to use to analyze the data. Could either be information like transcription factor binding sites or computations from systems biology models. So the three segments we saw, I'll just repeat this briefly. Errol showed us how metabolic allele classifiers can be used to distinguish AMR phenotypes and reveal underlying mechanisms, and how multiple, say, GWAS alleles that had been found map onto the same cellular process. And John has continued to expand pan-genome analysis, and in recent months and years, we have in particular made progress with virulence mechanisms that seem to come out of pan-genome analysis and are hard to get to in other ways. And then we saw Anand's pioneering work with the signal extraction ICA approach to help us modularize bacterial transcriptomes and really gain surprisingly deep understanding of them. And he has this bold goal to scrape all transcriptome data, bacterial transcriptome data he can get his hands on, and analyze it in a similar way, as he described at the end of his presentation. So thank you all for attending. I hope you enjoyed these three vignettes, and we'll be happy to take any questions you might have. Thank you for your attention.
Great. Thanks to all the presenters. This was a fantastic webinar. I really enjoyed it. I think Grace, we had mentioned earlier, if you have any questions, feel free to type them into the question box. While folks are thinking about them, maybe I could just ask a couple quick ones. And so this is really exciting stuff, also really complex stuff. So John, let me just start with you really quickly. In terms of your pan-genome, I thought it was really striking that you mentioned that genomes are doubling every year approximately. I assume that what drives the growth of a pan-genome in most of your studies is the accessory genome, not like the core genome. Is that correct? As these things grow? Oh, we might have lost. I'm going to unmute all our presenters.
Is John there?
He is, there he goes.
Yeah, I was muted by the organizer. So.
I'm sorry. Yeah.
So we see driving growth of the pan-genome. What happens with the core genome is a lot of times it starts to shrink, right? So we start to find maybe some sets of genes that are not found in a new novel strain, for instance. Yeah, was that your question? Yeah.
Yeah, I think so, and I think it was more or less along the lines of yeah, what's driving with the broader question, and then we can get to some other folks, is when genome's doubling, because of sequencing speed, what's the relative contribution of growth in the core genome versus the accessory genome? And I imagine these bacteria have been around for longer than a year ago, and so it's just interesting to me, like what are we finding out that we didn't know already from this rapid rate of increased sequence information?
Right. So it's a shrinkage of the core genome.
Okay.
Yeah, so the accessory genome continues to grow, and the core genome, if anything, would shrink, right? Because we may find a new strain that has a gene, there's missing a gene that maybe all our previous organisms had. But as far as in the growth of the accessory genome, a lot of things we're finding. I mean, sometimes we'll find it's really driven by evolution, right? So we may find a novel AMR allele, for instance, or a new gene that confers some kind of antibiotic resistance, or maybe potentially a novel virulence factor. And then the exciting thing about this too is it's not always just the presence of a gene like that, but also maybe the transfer of such a gene into a different strain, right? So maybe we find a gene that was never found in a clade of E. coli in the past. And so studying the presence and absence of these genes, their sequences, and even their distribution amongst the different strains, we learn a lot in every different scale. Yeah.
Gotcha. Thanks, John. All right. So I guess, Grace, do you mind reading off? We've got 3 or 4 questions in the question box. Can you go through them?
Sure. Yeah. So we have the first question is from Chen Liao, who is at the Memorial Sloan Kettering Cancer Center, and has a question about the metabolic allele classifier, and was specifically wondering about how metabolic flux state is determined for each isolate, since flux balance model would be undetermined without further data constraints.
That's a fantastic question. And so what we do is, so there are potential other solutions in that polytope that you can imagine, but we basically end up computing one and apply some penalties to try to minimize that space as much as possible. And so we have a harsh penalty or L1 normalization for the objective function. And so there's kind of few coefficients in the objective function actually determining basically what the network should be doing. And so there's kind of a smaller space of opportunity there, but it's true that there are other possible solutions as well that could also discriminate these strains. And so we compute a variety of solutions too, but the one that we find is within a small space in general, if that answers your question.
Sure. So Chen, if you have any follow-ups or if that doesn't answer your question, feel free. You can either raise your hand or you can ask another question. I can unmute you. But for now, we'll go on to Joe Campbell, who has two questions. One of which is, how good are they at predicting expression patterns during infections? And we'll start off with that, and then I can go on to the next one.
Wait, is this for the metabolic allele classifier?
I'm not exactly sure. Joe, if you wanted to ask your question in particular, I can unmute you, or let me see.
Yeah, Joe, we unmuted you, Joe.
Okay. I mean, I guess I was just interested in general because it seems to me that if you're thinking about using this to design therapies, you would want to know what kind of expression patterns you're actually going to see in a person who's infected with the pathogen.
Yeah, no, that's actually a great point. I was just asking because I guess also the question could be applied to Anand's transcriptomics as well. But yeah, for the metabolic allele classifier, we didn't actually check with transcriptomics data, so that's unclear, but would actually be a great way to validate these pathways further, basically just to test the consistency between basically high flux pathways and whether the genes in those pathways are expressed. So that'd be good validation, but I would expect that they would be.
And I guess if I'm still unmuted, I can just ask my second question is, how good is this as a potential diagnostic in the sense of if you were able to rapidly sequence an isolate that a person was infected with, and how good are you at guessing or determining whether the antibiotic resistance and sensitivity profile of an isolate from its sequence?
We could predict that very well, I'd say, even without the metabolic allele classifier, just pure machine learning with the genome sequences and resistant phenotypes have enabled us to basically identify these key alleles. So we could sequence that isolate, basically see if it has these K-AMR alleles, and if they do, we could classify it pretty well as either resistant or susceptible with high accuracy, probably up to the, yeah.
So how much have you, I mean, that's great. So how much have you done this explicitly by feeding this a genome sequence and that you know the resistance profile of and seeing what it comes out with? Does it always come out with the right answer?
It doesn't always. So it's, there's a accuracy. I don't have the exact numbers for how well it does, but we basically train the model on some training sets. So some genome sequences that it knows, and then we basically test it on these sequences that it's never seen before. And so for those ones it's never seen before, it does a pretty good job of classifying them. I'd say it classifies about 95% of them as correct for certain antibiotics. Yeah.
Am I on?
Yes.
Yes.
Did you have one more question?
Well, I was just going to maybe add to the answer here. We are desperately seeking in vivo expression profiles from infected animals to see how good the imodulins are in interpreting the difference between the in vivo and the in vitro environment, and in particular then to look at the difference between antibiotic treated animals and those that are not. And we're making some progress there. And John may say more, but we've been also looking at the predictions from the pan-genome analysis to look at virulence properties and how pathogens infect different patients. And he's identified, for instance, in E. coli UTIs, that there are about 10 genes that are always associated with the ability to cross the epithelial layer and cause bacteremia. So many of these methods are predicting valuable properties of pathogens in the host. Is there anything else you want to add, John?
I mean, I'll just harp on my point that, yeah, we believe that these methods are very valuable, and the more clinical data and phenotypic data we can collect for these different sequence strains, the more we can really test these methods in depth. So it's been a challenge. I think AMR data now is decently well structured and in the Patrick database, but other information related to clinical outcomes, et cetera, would be really valuable if it's associated with sequence genomes. So.
All right, so we have one more question from Chang. First of all, wanted to say thank you for the beautiful seminar from all of you, and had a question about the current structural characterization methods, stating that, for example, if it is possible to know some information of the structure, for example, the RNA secondary structure or protein structure of each gene sequence.
I guess I can start with that. So we've done some work to query the protein data bank, the PDB, where they have 3D structures of a lot of these proteins. And then there are methods, if you have a 3D structure, there are methods where you can attempt to adjust that 3D structure based on small mutations in the protein, and those vary in their accuracy and capability. But we have done a lot of that. If you're interested, you should check out some of our work. We have a package called SysBio, so it's structural systems biology, and we've actually pre-computed a lot of these modified 3D structures, and yeah, so I hope you check that out. I don't know if anyone else has an answer to that. Yeah.
Well, maybe I can just add a little to that. We have now structures for the 4100 protein in E. coli and how they assemble into complexes, and are now applying them in different ways. So the chemistry of ROS damage to protein is reasonably well known. So in one of John's slides, he showed the comparison between the structures of the pili protein of many different E. coli strains. And I believe it was the UTI strains that have many fewer ROS damage sites in the pili. So that's some correlation between structural information and lifestyle. And I believe we have some other ones, but they escape my mind right now.
All right, and then if there are no other questions, I think Reed had a question about imodulons.
I did. I have a very quick question, and again, naive, but maybe we can just go for it here. So, it was a really nice presentation on the imodulin, and I'm starting to understand it. I guess at a high level, are you surprised how similar different bacterial species imodulons are when you start to go back, outside of specific species of bugs, or was that expected?
So originally it wasn't, I wasn't exactly sure what to expect, but it seems like it's the fundamental properties, the fundamental functions that seem to be most consistent. For example, this translation imodulon that contains all the ribosomal subunits, I think every single organism that we've looked at has had this imodulon. Things like purine and pyrimidine biosynthesis are always in imodulons. So, and they always have the same gene. So it seems like this can also lead to evolution and evolutionary analysis of transcriptional regulatory networks as well.
Great. Well, thank you very much. Okay, well, I want to thank everybody again. If I don't see any more questions, you guys?
No, I do not.
All right. So, okay, great. Well, I want to thank the presenters one more time, and stay tuned for future webinars. And we look forward to hearing from the group. And I'm sure that reach out to presenters if you have any additional questions. So thank you all.
Thank you.
Thanks.
All right.